So if you look at question 9, okay, this question is from this year, okay, CHIJ paper, their CA1 paper, all right? And the previous questions that we have done, I'm sure you also can tell that the questions are all mostly from different schools, their CA1 papers this year, okay? So let's look at question 9 here. So substance K has a boiling point at 27 degrees Celsius. So this question, okay, what I'll do is I'll always draw what I call a number line, right? So for any substance, at a certain temperature, they will be in solid state, correct? At a certain temperature, they will also be in liquid state. At a certain temperature, they'll become gaseous state. So all the information that I know from this question is at 27 degrees Celsius, that would be substance K boiling point, which also means at 27 degrees Celsius and after 27 degrees Celsius, anything more, 40 degrees Celsius, 50 degrees Celsius, 28 degrees Celsius, okay, substance K will become gaseous state. Now, do I know what are their melting point or freezing point? Do I know at which temperature will they become solid state or liquid state? So far, I do not know, right? They only told us that substance K boiling point is at 27 degrees Celsius. So now if I look at part A, explain why substance K needs to be stored in a sealed container when it is placed in a room at 30 degrees Celsius. So what I'll do, right, is I'll ask myself, where is 30 degrees Celsius in this number line? So of course, 30 degrees Celsius is right here, somewhere here, right after 27 degrees Celsius. So this is where 30 degrees Celsius is. So again, anything after 27 degrees Celsius, we know that substance K is definitely in gaseous state. All right. So knowing that substance K is a gas at 30 degrees Celsius, why do you think I need to keep it in a sealed container? Now, your first thought will be, we know that gas will escape, right? It can basically move freely in the air. Okay, so that would be your common thought process here. But always remember, there needs to be a science concept behind this. Why do you think gas is able to escape and move freely in the air? Why? Okay, so you cannot just tell me, okay, substance K will not escape because it's in gaseous state. That's not enough. So over here, we need to recall something, okay? We need to recall the topic on matter, okay? And the property, basically, of gas, right? What is the property of gas? How come gas is able to move freely in the air and just escape like that? Okay, it's very hard to contain it. That's why it needs to be sealed. So ask yourself, okay, what's the property of gas? Gas in this case is, we know that it does not have a definite shape, it does not have a definite volume, and it also can be compressed. Now, so we got to eliminate here, right? Which one are the ones that are not relevant here? Okay, the reason why I want to put it in a sealed container is because I don't want gas to escape, right? So in this case, do you think it has to do with gas not having a definite shape or not having a fixed shape? No, right? This is not about it being able to take up the shape of any container, okay? Neither can it be compressed. I'm not actually adding in more, right, into a fixed container, okay? I'm not adding in more gas into the fixed container. So the reason why I need to contain it is because gas does not have a definite volume. That's why if I open it, okay, gas can actually move freely in the air. Okay, simply that. Which is why at 30 degrees Celsius, the first thing I would say is K is a gas, okay? You need to identify the state of it at 30 degrees Celsius. So yes, it is a gas. Secondly, why must it be sealed? Because it does not have a definite volume. And finally, you link back, which is why it will escape to the surroundings if the container is not sealed. Okay? So that's your answer for part A, right? So you can see it's a combination of heat and matter topic as well. So you got to be very, very familiar with your concepts here. All right? So now let's move on to B. Okay? Using the above information, would it be possible to tell the state of substance K at 0 degrees Celsius? Would it be possible to tell what state substance K will be at 0 degrees Celsius? Now, 0 degrees Celsius, again, if we look at the number line, right, is way below 27 degrees Celsius. But again, like I said just now, what is the information that has been told to me, right? The information is Ki I only know K's boiling point. Do I know the melting point? Do I know the freezing point? No. Do I know exactly which temperature will it start to melt or freeze? Again, no. So it is not possible to tell in this case, right? Because you have to say the melting or the freezing point, okay, of K is unknown. I do not know what the melting or freezing point is. Now, bear in mind, let's do a bit of recap. Remember that melting point and freezing point are the same, okay? So let's say I tell you the melting or the freezing point of K is maybe at 15 degrees Celsius. So 15 degrees Celsius means what? If it's at 15 degrees Celsius, at 0 degrees Celsius, I definitely know it will be solid state, right? Okay, if I tell you that the melting point is also at 15 degrees Celsius, would I know which state will it be? Liquid? Yes, anything at 15 degrees Celsius and more than 15 degrees Celsius, right? So between 15 and 27. Of course, that is just an example, okay, for me to identify whether it will be a solid state, liquid state, okay, solid state or liquid state. So in this case, I do not know the melting or freezing point. That's why I will not know what state K will be at 0 degrees Celsius. Now, let's move on, all right? This is a common question that they like to ask. Okay, so again, they combine this with water cycle. Okay, so here you have Dilys that compared the rate of evaporation of liquids P, Q, and R. She poured 10 cm cube of each liquid into 3 similar glass beakers and placed them by the window in a science laboratory. She noted the time taken. Now, once she noted the time taken, I know that this would be my measured variable, right? I am measuring the time taken for each liquid to evaporate. Okay, and again, what do you think is my change variable? So previously at the first sentence, we already mentioned this. There are liquids P, Q, and R. So my change variable, to be more specific, remember you cannot just say liquids P, Q, R, all right? So liquids P, Q, R means what? What is the variable here? So in this case, it's the type of liquids, okay? So as long as I see different liquids, my variable will be the type of liquids here. Again, my measured variable is the time taken for each liquid to evaporate. So very simple 3-mark question for part C, okay? Based on the table, right? Based on the time taken for each liquid to evaporate, which one do you think evaporated the slowest? Of course, it took the longest time, right? So in this case, it's 51 minutes, which is your Q, okay? Followed by P, and the fastest one, the one that took the fastest time to evaporate is, of course, your liquid R. Okay, 3-mark question, simple. Another common question they love to test, okay, is part D. All right, an experimental setup question. All right, if you have been following our YouTube channel, okay, we do have a video on these experimental question types, like your fair test, reliable test, okay, conclusion questions, relationship, all right, control setup, okay? So this is one of them that they love to test nowadays, okay, very, very commonly tested on fair test questions. So since it's a June holiday program, right? Okay, we have also input a lot of writing structures that are crucial, okay, very important, that will really help you to target and answer these questions accurately. So if you look at part D, okay, of course in exam, you will not see this writing structure here. So this, because it's a revision course, okay, we are here to help you, right? So this is your writing structure here, which will help you answer part D accurately, okay? So if you look at part D, they say, give a reason how using similar beakers helps to make her experiment fair. So before you tell me your answer, okay, don't be too quick to tell me that, okay, similar, the beakers must be the same, must be similar, right? Because there can only be one change variable in the experiment, which is a type of liquids, DN. Okay, that's not going to get you any marks, all right? Usually for this kind of question, it can range between 1 mark or 2 marks, okay? Give a reason. You are to explain the target of this variable here. And the main part here is, why do I need to use similar beakers? Why is it so important to use beakers of similar sizes? Right? Yes, I know it's going to affect my measured variable if I don't use beakers of same sizes, but you need to explain further. If I use beakers of different sizes, what am I going to affect? What variable will I change which will then affect what I am measuring, which will then affect the time taken for each liquid to evaporate. All right. So if you look at my writing structure here, I said keeping variable P, okay, similar beakers or whatever variable they give you, let's call it variable P, okay? So keeping similar beakers the same, right? Why must I use beakers of the same size? You realize that there is this variable that I called, okay, I called it as variable Q. Now, this is the part that is very, very challenging here, okay? Which is why they love to test this question, because this is the more challenging ones, all right? But to keep it simple, what is variable Q? The moment you're able to identify variable Q, everything else is very, very easy, okay? So what is variable Q? So like I said earlier, if I were to change the size or the shapes of the beakers, if I use different beakers of different sizes, okay, what will I affect? That would be my variable Q, right? What will I affect? Which will then affect my measured variable, which will definitely affect the time taken for the liquids to evaporate. So you have to think a little bit deeper, okay? So first thought process, you ask yourself, what if I now have beakers of different sizes? So don't know, just draw, right? Imagine I have this beaker here, one size that looks like that, and another one that maybe is very tall, okay? Let's just use two, okay? Keep it simple. And now, how much water must I pour? How much liquid must I pour? 10 cm cube of liquid, right? So now let's say I have the same amount of liquid, but you know that if I pour in this, the first beaker, okay, the water will probably be like that, right? If I pour the same amount of water or same amount of liquid into this container, it'll probably be higher, right? Maybe like that. So what am I changing here? Okay, what is variable Q? What am I changing here? Which will then affect the time taken for each liquid to evaporate, right? So in this case, if you look at this, you know immediately what are things that affects the rate of evaporation. If you look at this, I am changing the exposed surface area of the liquid, isn't it? Right? You see, the liquid exposed surface area are different. And we know that the exposed surface area, whether it's larger or smaller, it will definitely affect how fast the liquid is going to evaporate. It's going to affect the rate of evaporation. That is my variable Q. So variable Q in this case is simply the exposed surface area of the liquids. So once you identify that, everything just follow our writing structure and you will get your answer, okay? You don't believe me? Look at variable P. Variable P again is just the question, whatever the question is asking for. Why must I use similar beakers? Okay, variable Q is what we've identified. Measured variable is given in the question. I've already stated here. The time taken for each liquid to evaporate completely. Okay, and the last sentence is you want to ensure, like what you said earlier, that there can only be one change variable. And what's our change variable? We have already identified earlier. So once you gather all these and once you use our writing structure, you got your answer. Okay, you just need to put everything into this writing structure and here you go. Right? So instead you can say using, you can say keeping similar beakers, okay, or in this case using similar beakers would ensure that the exposed surface area of each liquid, and I'll explain further to say in contact with the surrounding air, right, would be kept the same. Okay, and would not affect, of course, don't copy measured variable, okay, you must identify it, which will affect the time taken for each liquid to evaporate completely. And then finally, this would ensure that there will only be one change variable, which is, again, identify the change variable for clarity, all right, which is the type of liquids, okay, that would affect your results here. Okay, so you just need to expose yourself to more of this question. Every time you see similar type questions, just use the writing structure. Think a little bit deeper. What is variable Q? What if I change variable P, right? If I change variable P, how am I going to affect variable Q? Okay, right? What will variable Q be? What am I going to affect, right? Which will then affect my measured variable. So that should be what you need to think of a little bit deeper. And remember, it's always your science concepts, okay? Whatever you have been taught. All right? Now, question 8. Okay, this is a weighted assessment paper, all right, from this particular school, okay? So I call this more or less testing on your life processes, things like photosynthesis, respiration, all right? So another common question they love to test, right? So if you look at this, liquid A is used to test for the amount of carbon dioxide, okay? The diagram below shows the change in the colors of liquid A in the presence of different amounts of carbon dioxide. So you can see here that this liquid A, all right, according to the amount of carbon dioxide, it will change color. So we know yellow has a lot of carbon dioxide. It detects a lot of carbon dioxide. Later on, you will see that it's in this in this water, in this beaker of water, okay? And of course, it will turn blue if it detects very little carbon dioxide inside the water, okay? So this is the whole color trend, right? How it changes. So if we read on, Macy added 2 drops of liquid A in a beaker filled with water. She then blew gently into the beaker using a straw, okay, until the liquid turned yellow. So if you look at yellow, oh, okay, you should know why, right? Why did she blow gently into the beaker such that the liquid turned yellow? Yellow means there's a lot of carbon dioxide, okay, and she blew it because, of course, she is, in a way, carrying out respiration, right? So when she's respiring, of course, when she blows out, she gives out, okay, carbon dioxide. So she's providing carbon dioxide or adding carbon dioxide into the beaker of water, okay, making sure that the liquid turns yellow first. So there's a lot of carbon dioxide inside, okay? So the beaker was covered with a piece of glass after a hydrilla plant was added. So you see a sun here, you see the hydrilla plant. What is the first thing you need to identify? What do you think it is carrying out? Okay, and earlier she has really blew into the beaker. So she provided the water with a lot of carbon dioxide, and all the more there's light. So we know that this hydrilla plant, okay, is carrying out photosynthesis, right? It's making food, okay? So let's read the question for 2 marks, okay? Explain why the liquid in the setup turned dark green. Now, let's look at dark green after 30 minutes, right? Dark green, we know that, okay, it seems like the amount of carbon dioxide here is low. Okay, the amount of carbon dioxide in the water is low. Okay, so why do you think this liquid A would detect very little carbon dioxide left in the water after 30 minutes? Okay, so first we need to identify something. Again, what is this hydrilla plant doing? Okay, number one, we know that it is in the presence of light. So when it's in the presence of light and there's carbon dioxide in the water, it will carry out photosynthesis. So a lot of times, I know you are all able to identify the first part. You will say, okay, the hydrilla plant is going to take in carbon dioxide in the water, right, which is given out by Macy, okay? And always identify your life process, all right? Very important to identify this, and will carry out photosynthesis in the presence of light to make food for itself, right? And allowing it to give out oxygen. Okay, so the main thing here is telling me that it will take in carbon dioxide in the water. It will carry out photosynthesis in the presence of light. Okay, so what is happening here? We know that it's taking in carbon dioxide, right? So of course, the level of carbon dioxide in the water would decrease. Now, if you stop here, okay, this is another common issue that a lot of students tend to make. Okay, what is the error here? The error here is what? You don't answer the question fully. Okay, by just telling me that the hydrilla plant will take in carbon dioxide, carry out photosynthesis, okay, in the presence of light, doesn't tell me, okay, you have not answered the question yet. It doesn't tell me why then will the liquid turn dark green. Okay, so again, linking back. Okay, you need to link back to the question, which is why a lot of times read your answer, read the question again, and ask yourself whether you have answered the question accurately. All the more, this is a 2-mark question, right? So why then, okay, will the liquid turn dark green? So your next mark come from the linking back part. So you must say, because your plant is taking in carbon dioxide in the water, okay, the amount of carbon dioxide will decrease in the water, right? Which is why liquid A will turn dark green. And we know from the previous table, when the amount of carbon dioxide in the water is low, dark green or blue is a possibility, right? So this part here is so important, telling me that the amount of carbon dioxide in the water would decrease, okay? And therefore A would turn dark green after 30 minutes.